Welcome to The Explainer. Today, we're going to unlock one of the most fascinating mysteries happening right inside your own body. Think about this for a second. The cells that make up the hair on your head and the soft tissues of your liver came from the exact same blueprint. We're going to explore how a single set of biological instructions can build everything from those hair follicles to the lining of your stomach. It is an incredible story of microscopic engineering, and it all starts with a very specific, honestly puzzling paradox. Okay, let's dive right in. So here's the central question we're exploring today. Aside from a few rare exceptions like gametes or random mutations, almost every single cell in the human body has the exact same sequence of DNA. Your liver cells, your skin cells, your brain cells, they all share the exact same genetic code. So how on earth do they end up looking and behaving completely differently? Section 1. The Grand Cellular Paradox. To solve this grand mystery, we first need to look at the sheer scale of the information packed inside us. Humans have roughly 20,000 genes in our DNA. Just try to imagine a massive, dense database of information packed into a space smaller than the head of a pin. That's exactly what exists inside nearly every single cell of your body. These 20,000 genes code for every single biological function that keeps you alive, breathing, and healing. But remember the core of our paradox here. This entire, massive database is present everywhere. Now, consider just how wildly different these cellular jobs actually are. Liver cells are handling super complex metabolism, breaking down toxins and processing your nutrients. Hair cells, they're building a tough physical structure that can withstand the elements. Brain cells are firing these rapid-fire electrical signals to process your thoughts. And skin cells are acting as a physical shield to protect your internal organs. It's almost absurd, right? All these wildly different cells with completely different shapes and functions are somehow running on the identical set of 20,000 genetic instructions. To understand how they specialize to perform such diverse jobs, we need to uncover our first foundational clues. Section 2. The Foundational Clues, the Central Dogma. Let's take a look at the basic molecular machinery of life and see how information actually moves inside a cell. This three-step flow of information is basically our roadmap. Information is stored in DNA, which acts kind of like a stable long-term archive, securely locked away in the nucleus. That information is then transcribed into a temporary disposable copy called messenger RNA, or mRNA. Finally, that mRNA is translated into proteins, which go out and do the actual work. Now, you might occasionally hear people claim that this central dogma has been debunked because scientists have found a few minor exceptions over the years, but honestly, that's really just a semantic debate. While it might not be a flawless rule for every obscure biological scenario, this information flow remains an extremely useful, powerful model for understanding how your cells operate day in and day out. The difference between those first two steps is absolutely crucial. DNA is double-stranded and uses the bases adenine, thymine, cytosine, and guanine, A, T, C, and G. It basically just sits there safely and stably storing your genetic information over a lifetime. But when a cell needs to use a specific piece of that information, an enzyme called RNA polymerase swoops in and temporarily unzips the DNA. It creates a single-stranded mRNA copy. Notice how mRNA swaps out the thymine for uracil, or U. This mRNA is semi-transient. It gets created, floats around the cell to carry the message to the factory floor, and eventually breaks down once its job is done. This whole process we're talking about has a specific name: gene expression. When we say a gene is being expressed, it means the cell isn't just passively storing that tiny piece of the blueprint, it is actively reading it and creating a physical product, like an mRNA strand or a protein, that floats around and actually performs a function. If a gene isn't expressed, it's just sitting idle in the database. And differences in gene expression? Well, that's the very key to our paradox. Section 3. Translating the code. Let's see how this intermediate mRNA actually builds the physical workers of the cell. So the really crucial point is how the cell actually reads the mRNA. It doesn't read it one individual letter at a time. The mRNA strand attaches to a cellular machine called a ribosome, which reads the sequence in chunks of 3 letters at a time. We call this triplet of letters a codon. Think of it kind of like reading words in a sentence, where every 3-letter word tells the machine to grab one very specific building block, an amino acid, and add it to a growing chain. I really want you to take a second to appreciate how sensitive this translation process is, because the code is read in these precise 3-letter codons. A single 1-letter difference in your DNA sequence changes the resulting mRNA codon.
That single tiny change can swap out an amino acid, which can completely alter how the final protein functions in your biochemistry. It is incredibly precise, and it's exactly why tiny genetic mutations can have such profound effects on our biology.
Once that chain of amino acids is strung together, it intricately folds into a 3D structure to become a functional protein. Proteins are the actual workers of the cell. They do the heavy lifting, running your metabolism, holding the cell together, sending signals. But look at that last job, transcription factors. These are special proteins whose entire purpose is to turn around, bind right back to the DNA, and regulate the expression of other genes. It creates this massive, complex, interconnected network of genes turning other genes on and off. It's amazing. But even with this incredible regulatory network, we are still missing one massive puzzle piece to fully explain our original paradox.
Section 4, the epigenetics reveal. Okay, we are finally ready for the twist that solves the mystery of why identical DNA creates totally different cells.
Enter the absolutely mind-bending world of epigenetics. It turns out your biology isn't just about the linear sequence of the A, C, T, and G letters. It is heavily dependent on how the massive DNA molecule is physically organized and folded in three-dimensional space inside the cell. It's not just what the code says, it's what shape the code takes. Now, as much as biologists love their sheet music metaphors, the whole DNA is the sheet music and epigenetics is the musician playing the song thing, those cliched metaphors can honestly make things more confusing. We don't really need metaphors today. The literal, physical reality of what is happening to our chromosomes in 3D space is way more fascinating on its own. Let's look at the mechanics. Picture zooming in on that physical structure. Inside the nucleus of the cell, your DNA isn't just floating loosely. It is carefully wrapped around these specialized proteins called histones. If the DNA is loosely wrapped, that section is physically accessible. The RNA polymerase can easily swoop in, read the code, and express those genes. But if a section of DNA is tightly coiled around those histones, it is physically blocked off, totally inaccessible to the cellular machinery. Any genes hidden in those tight coils are effectively turned off or silenced. The physical coiling literally dictates which genes a cell can even attempt to use.
Section 5, becoming a specific cell. Let's bring all these clues together to answer exactly how our bodies build different tissues from the exact same blueprint.
So this entire process is called cell differentiation. In the very beginning, an embryo is just full of general, unspecialized stem cells that don't have a specific job yet. But as they develop and multiply, these cells adopt unique epigenetic regulatory profiles. That means the DNA in a future brain cell physically folds and coils completely differently than the DNA in a future liver cell. In the liver cell, the genes required for metabolism are left loosely wrapped and perfectly accessible, while the genes for making hair are tightly coiled and permanently silenced. In a hair cell, the exact opposite happens. It's this unique 3D folding that gives cells completely different biological roles, even though their raw DNA sequence is a 100% identical match. Which leaves us with one final pretty profound thought. We know that cell fate is determined by epigenetics, by which specific genes get coiled up and which get left wide open. But the actual origin of this mechanism, how the embryo knows exactly which sections of DNA to coil and uncoil in those very first stem cells right at the beginning, remains one of biology's greatest ongoing mysteries. As we continue to decode the physical structure of our DNA, we get closer to understanding the very origins of our biological destiny. This is literally what makes you, you. Thanks so much for joining me for this explainer, and keep questioning the incredible science happening inside your own cells.